everyone, this is Sarah Barth. I chose to do a case study on open-angle glaucoma. So open-angle glaucoma is the case, and it's caused because of mutations in a gene on chromosome 1. When fluid's not able to drain out of the eye, the fluid stays there and accumulates so much that it can end up blocking peripheral vision. Eventually, this disease is progressive, and it's usually by the age of 40 that the vision is affected. I chose open-angle glaucoma case because I wanted to choose something that I knew very little about. I've heard of glaucoma before, but didn't really know anything about it, and I didn't even know that there were different types of glaucoma. So the mutations in the GLC1A gene that cause open-angle glaucoma is mutations from tryptophan to arginine, glycine to valine, and tyrosine to histidine. So for the mutation of tryptophan to arginine, if you look at this genetic code, a codon change that would explain the amino acid tryptophan being substituted for arginine would be changing the codon from UGG to CGG, changing that U to C. The other mutation from substituting valine for glycine could be because of substituting that second letter. If there was a change in the codon, changing the second nucleotide G to U, because you could see there that GGU, GGC, GGA, or GGG all make glycine. So changing that second codon to U would create valine instead of glycine. Again, you can go back and see on this chart that those make that amino acid. So the other mutation would be from tyrosine to histidine, and for this one, changing that first letter from U to C would create valine instead of glycine. So there are mutations in this gene that would not alter the amino acid sequence. By making certain substitutions and having there be mutations, the amino acid would still stay the same from proline or threonine or glutamate. So for proline, if any of these nucleotides are changed, it would still stay proline. So if you look there, the CCU, CCC, CCA, and CCG all make proline. And if you change just a little bit of the letters, so if you change the third nucleotide from U to C or A or G, it's still proline. And the same thing here with threonine, changing that third nucleotide makes no change in the amino acid. It would still stay threonine. And glutamate, if we were to change GGA, change that third nucleotide to G, or the other way around, if you had GAG and the third nucleotide was substituted from G to A, there would still be no change. And this is because of what's called a mutation called polymorphism, which is variants in the DNA that sequences that coexist in population. These are with the higher frequency mutations, and they make no change to the phenotype. So a gene that, when a mutation to this gene that can cause open-angle glaucoma might encode a protein that participates in apoptosis, which is a programmed death of cells. So the RGC cell, when this one goes through apoptosis and you have RGC cell death, it causes that permanent vision loss in glaucoma. So this apoptosis is the end of RGC death progression glaucoma. There could be a lot of different things that start the process of apoptosis in the RGC. One of those things could be the neurotrophil deprivation. And the way that this case comes together with the class is a lot of things, almost everything that we've done from the beginning of the class, starting from the first week with DNA structure, and like you've seen in those other screens with the DNA sequencing, protein encoding, and we've talked about apoptosis in week 3, and then in week 2, and then week 3 we started talking about mutations, which is how this disease progresses or it happens is because of mutation in the cell. And something that I found interesting about this case was that the glaucoma actually affects more women than men. About 55.4% of all cases of open-angle glaucoma were women. And something else that I found interesting is that Europe has the most cases of open-angle glaucoma. It is mostly in, and also then after Europe, it would be China and then India. Right, thank you.